Welcome back to the lab, everyone. This is the Deep Dive Lab. And actually, before we jump in today, I have to ask you something kind of personal.
Oh, boy. Yes, you.
So, what happens to your stress levels when, your phone buzzes with a notification and it's from your extended family group chat?
Oh, immediately spiked.
Yes.
Just completely spiked. It's like this Pavlovian response. I see the group name and my heart rate just goes right up.
Right, exactly. It's so universal. I mean,
Yeah.
you've got that one uncle, let's just call him Uncle Bob, who types exclusively in all caps.
Oh, yeah.
Screaming about some news article that he definitely didn't actually read.
Never reads them.
Never. And then you've got the cousin sending these cryptic emojis that could mean I'm happy or I'm in jail.
Right,
Huh?
totally unpredictable.
And then, thank goodness, you have the peacemaker aunt who, steps in with a casserole recipe or heart emoji to just talk everyone off the ledge before Thanksgiving gets canceled.
It is a very delicate ecosystem. I mean, if the peacemaker ant takes a nap, the whole chat just implodes.
It really does. Well, the reason I bring this up is because today we're going to find out that this exact drama, the screaming, the confusion, the de-escalation, it's all happening inside your body right now.
That is actually a frighteningly accurate comparison.
It's not on WhatsApp, it is in your bloodstream. We are taking a deep dive into the body's text messages, specifically the interleukin-10 family of cytokines. And honestly, this family has more drama than an episode of Succession.
They really do. And, for our listeners, cytokines are the signaling proteins of the immune system.
Yeah.
They literally are the text messages.
Right.
They tell your cells to grow, to die, to fight a virus, or to just chill out.
Right.
And the IL-10 family is so special because it covers that entire spectrum, from healing to killing to calming.
And to guide us through this molecular soap opera, we are looking at a really heavy hitter of a research paper today.
We are.
It's titled Structure and Function of Interleukin-22 and Other Members of the Interleukin-10 Family.
Yes, this is a major, really comprehensive review paper.
Mm-hmm.
We definitely need to shout out the lead author, Daniela Barreto Barbosa Trivella, and the senior author, Igor Polikarpov.
Mm-hmm.
They are from the University of São Paulo. And they collaborated on this with Jean-Christophe Renaud from the Ludwig Institute for Cancer Research.
What I really love about this paper, and the reason we chose it for the Deep Dive Lab today, is that it doesn't just tell us what these immune text messages say, it actually tells us what the letters look like.
Right.
It's all about the physical shape.
Exactly. Because in biology, shape is destiny. If you don't understand the geometry of the key, you are never going to understand how it opens the lock. And Trivella and Polikarpov, they really get into the nitty-gritty of these atomic structures.
So our mission for this deep dive is to decode these shapes. We are going to meet the Peacemaker, the Double Agent, and the Ghost.
I love those nicknames. It makes them sound like an Ocean's 11 crew.
I try my best. So let's unpack this. Who is actually in this biological family tree?
So, looking at the Trivella paper, they categorize this family into three main branches. First, right at the head of the table, you have IL-10.
The Peacemaker.
Yes, you called it the peacemaker, and that is scientifically spot on.
The Aunt Linda of the immune system.
Exactly, Aunt Linda. Then you have a group we call the IL-19 subfamily, and this includes IL-19, IL-20, IL-22, and IL-24. We often call them the skin squad because they are very, very active in tissue repair and skin health.
Okay, and the third group, the cousins who show up uninvited.
That would be the interferon lambdas, or IFN lambda. Think of them as the Border Patrol.
Border Patrol, got it.
They are technically cousins, distantly related, but they use the same door to get into the house, so structurally they are part of the family reunion.
Okay, I want to visualize these things. I'm looking at the diagrams in the source material right now, and to me they look like, bundles of twisted pool noodles.
Pool noodles is actually a really good starting point.
Yeah.
Scientifically, we call them helical cytokines. So imagine you have 4 short pool noodles. Let's say a blue, a red, a green, and a yellow one.
I'm picturing it.
Now grab them in a bundle and give them a twist. That is a 4 helix bundle. In the paper, they label these helices A, C, D, and F. This bundle is the core of the message. It's the package itself.
But it's not just a straight bundle, right? Because I'm looking at helix F in the diagram, and it looks like someone snapped the noodle.
Ah, put a pin in that, because that is the most critical detail. The paper highlights this structural quirk, and they call it the kink.
The kink.
Yes, specifically in helix F. Imagine taking that pool noodle and bending it, kind of like an elbow. It's not broken, but it's definitely bent at a sharp angle.
Why does a bent pool noodle matter so much?
Because that bend is what exposes a very specific electrically charged part of the molecule. It creates a hot spot,
Oh, I see.
like sticking your elbow out to ring a doorbell. If the noodle was totally straight, that charged area would just be hidden inside the bundle. But because it's kinked, it's exposed.
So if the kink isn't there, the text message never gets delivered.
Exactly. The receptor on the cell wouldn't even recognize it. It would just bounce right off. That tiny structural oops is actually the entire reason the whole system works.
Wow. Okay, so we've got the message, the kinked bundle of noodles. Now it has to be received. I read about this lock and key mechanism in the paper, but honestly, it sounds more like a heist movie where you need two keys to open a vault at the same time.
That is a much better way to think about it.
Yeah.
These are class 2 cytokine receptors.
Yeah.
It is not a simple doorbell. For the signal to actually go through, our cytokine, the text message, has to float up to a cell and grab onto 2 different receptor chains.
Okay, so it grabs receptor 1.
Right, let's call it R1. But just grabbing R1 does absolutely nothing.
Really?
Yeah, the cytokine has to hold onto R1 and then literally reach over and grab R2. It physically bridges them together.
Oh, so it pulls them in for a group hug.
Exactly.
Yeah.
And only when R1 and R2 are physically touching does the signal fire inside the cell.
And that signal, this is where we get into the alphabet soup, the JAK-STAT pathway. I see this in every medical paper and my eyes usually glaze over. Can we make this a bit more interesting?
We definitely can. Think of it like a corporate office. The receptors, R1 and R2, are the receptionists at the front desk. When they get brought together by the cytokine, they call the managers in the back office.
Okay.
Those managers are the JAK-STAT proteins.
Managers are awake.
Right, so the JAKs then tag a courier service, the STAT proteins. These STATs grab the memo, sprint through the office lobby, which is the cytoplasm, and go straight into the CEO's office, the nucleus.
And what do they do in the CEO's office?
They slam the memo on the desk and rewrite the company policy. They literally bind to your DNA and change which genes are being turned on or turned off.
So the text message comes from outside, says there's a virus. The cytokine bridges the receptionists, the managers wake up the couriers run to the CEO, and the CEO starts printing weapons.
That is the JAK-STAT pathway in a nutshell. It is an incredibly fast, efficient relay race.
And the crazy part is that the shape of the pool noodles determines which receptionist they can grab, which then determines which memo gets sent to the CEO.
Precisely. Shape dictates everything.
So let's look at how that shape changes the personality of these family members. Let's start with the head of the family, IL-10. Why is Aunt Linda the peacemaker?
Because without IL-10, you would likely die from your own immune system.
Wow. Okay, stakes are very high.
Extremely. When you get an infection, your body sends out attack signals, things like TNF-α, that's Uncle Bob, screaming in all caps. You need that inflammation to kill the bacteria.
Right.
But if that screaming never stops, you get massive collateral damage. That's how you get autoimmune diseases like colitis or rheumatoid arthritis.
So IL-10 is the mute button.
It is the master regulator. It tells the cells, okay, the war is over, put down the weapons. But here is the real wow factor from the Travella and Polikarpov paper. IL-10 does this because it is shaped completely differently from its cousins.
Wait, I thought they were all bundles of noodles.
They are. But almost every other member of this family, like IL-19 or IL-22, they work alone. They're monomers, just one single bundle floating around.
And IL-10.
IL-10 forms what the paper calls an intimate intertwined dimer.
That sounds very cozy.
It is. Imagine 2 of these 4-helix bundles hugging. But it's not just a polite church picnic hug. They are hugging so tight that they literally swap limbs.
Wait, they physically swap parts of themselves?
Yes. Structurally, the helix F of one molecule reaches over and inserts itself into the bundle of the other molecule, and vice versa with helix E. They form this V shape. They become one single functional unit. You literally cannot separate them without destroying the protein.
Why does nature go to that much trouble? Why not just have 2 separate ones?
That V shape creates 2 distinct binding sites on opposite sides of the V. This specific geometry allows IL-10 to bridge its receptors in a very stable, high-affinity way. It essentially puts a bear hug on the receptors that send a very strong, sustained, calm-down signal to the cell.
So the structure, the literal hug, is what saves us from chronic inflammation.
Structure is function. And this is exactly why researchers are obsessed with IL-10 for treating things like lupus and rheumatoid arthritis. They want to harness that V shape to calm the immune system down when it's throwing a tantrum.
Okay, so IL-10 is the benevolent, hugging peacemaker. Now I want to completely flip the script. Let's talk about the member of the family that acts like a character in a spy thriller. The double agent, IL-22.
Ah, IL-22. The Jekyll and Hyde of the family.
Structurally, this one is a loner, right? No hugging.
Correct. It is a monomer. It works alone.
Yeah.
And depending on where it is working in the body, it can either be a superhero or a villain.
Give me the superhero version first.
In the gut and in the lungs, IL-22 is incredible. It promotes tissue repair. So if you have damage to your gut lining, maybe from a bacteria or just really spicy food, IL-22 tells the cells to proliferate. It says, rebuild the wall.
That's great.
It even tells them to produce beta defenses, which are basically the body's natural antibiotics.
So it's like a construction crew showing up to fix a pothole on the road.
Exactly. We love IL-22 in the gut.
Yeah.
But imagine that same construction crew shows up on your skin where there is no pothole,
Yeah.
and they just start pouring concrete anyway.
And that's the villain arc.
That is psoriasis. Psoriasis is essentially skin cells that are growing way too fast and not maturing properly. So they pile up into those red scaly plaques.
And IL-22 is the one shouting, grow, grow, grow.
Along with its cousins IL-19 and IL-20, yes. The source paper explicitly notes that IL-22 induces hyperproliferation of keratinocytes, which are your skin cells.
It's like a gym trainer who is great when you need rehab for an injury, but then just keeps screaming at you to do squats until your legs completely give out.
That is the perfect analogy. In the gut, you need rehab. In the skin, you need rest. But IL-22 is just screaming squats.
So here is the million-dollar question for you. How does the body know to send the construction crew to the gut but keep them away from the skin? Or rather, why does the message get so confused in psoriasis?
It comes back to the keys. This is a massive discovery in the Polikarpov paper. IL-22 shares one key with the peacemaker IL-10. The receptor, IL-10R2. That's the generic key. But its second key is totally unique. It's called IL-22R1.
And where do we find IL-22R1?
That is the trick. It is found almost exclusively on tissues: skin, pancreas, gut, lungs. It is not found on immune cells.
Wait, back up. That seems weird. Usually immune signals talk to immune cells, like white blood cells talking to white blood cells.
Not this one. IL-22 is an immune signal that completely ignores the police force and talks directly to the buildings. It doesn't tell the army what to do. It tells the walls to get thicker.
That is surprisingly specific. It's like having a radio frequency that only the bricklayers can hear.
Precisely. And knowing that receptor exists on the skin allows scientists to try and block it. If we can design a drug that gums up the lock on IL-22R1, specifically in the skin, we can stop the growth signal of psoriasis without shutting down your entire immune system.
That is incredible precision. Okay, moving on to the character in this family that I genuinely think deserves its own movie franchise, IL-24.
The ghost.
I call it the Ghost because the more I read about it in the source material, the spookier it gets. It was originally named M-7, which sounds like a secret weapon code. What does that actually stand for?
Melanoma differentiation associated gene.
So it was discovered in skin cancer.
Yes. Researchers were looking at melanoma cells, which are just skin cells gone rogue, and trying to force them to differentiate or turn back into normal behaving cells.
All right.
They found that when this gene, M-7, which we now know as IL-24, was turned on, the cancer cells just died.
But here is the magic bullet part that completely blew my mind. It kills cancer cells, but it completely ignores normal cells.
It is the holy grail of oncology. It induces apoptosis, which is programmed cell suicide, in tumor cells, but it leaves healthy cells totally untouched.
How does a protein know the difference between a good citizen and a bad actor?
That is the question driving so much research right now. The Travella paper suggests IL-24 works in two ways, and this is where your ghost nickname really fits perfectly.
Really got me.
First, it can work like a normal cytokine. It floats outside the cell, hits the receptors, and sends a signal. Hey, launch the missiles.
Standard outside attack.
But there is an intracellular isoform, a version of the protein that stays inside the cell.
The call is coming from inside the house.
Exactly. It accumulates inside the cancer cell's factory, the endoplasmic reticulum, and it starts pulling wires. It targets the mitochondria, the power plants, and causes massive stress. We call it ER stress.
So it literally sabotages the machinery from the inside.
It creates a highly toxic environment of reactive oxygen species. Now here is the catch. Normal cells have safety valves. They can handle a bit of the stress, or they have enzymes that can degrade the protein before it does real damage.
But cancer cells can't.
Cancer cells are already running their factories at 110% capacity. They're stressed out just by existing. When IL-24 adds this extra stress, it pushes them over the edge, they overload, and they shut down.
That is wild. It exploits the cancer's own greed for growth against it.
And that's exactly why the paper discusses clinical trials using adenoviruses.
Using a virus to treat cancer? That sounds like the plot of I Am Legend.
Hopefully with a much better ending. But they essentially use a modified cold virus as a delivery truck. They pack the virus with the IL-24 gene and inject it directly into the tumor. The virus infects the cancer cells and starts printing out this ghost protein inside them. It's a molecular grenade.
It really highlights how beautifully diverse this family is. You have the V-shaped hugger, IL-10, keeping the peace, the overenthusiastic gym trainer, IL-22, building walls, and the ghost assassin, IL-24, taking out the bad guys.
And we can't forget the cousins at the border.
Right, the interferon lambdas, the border patrol.
These are the newest members to the family reunion.
Now, I have to say, when I hear the word interferon, I shudder a bit. I think of old-school hepatitis C treatments where people felt like they had the flu for a solid year, fever, chills, absolute misery.
That flu-like feeling is very real. The reason standard interferons make you feel terrible is that their receptors are found everywhere. Your brain, your muscles, your blood vessels, when you take them, your whole body thinks it's under attack. It is a full system lockdown.
But IFN-λ is different.
Structurally, the paper classifies it in the IL-10 family because it uses that same master key receptor we talked about, IL-10R2. But its second receptor is highly specific.
Specific like IL-22.
Even more so. The receptors for IFN-λ are found almost exclusively on epithelial surfaces, the linings. The lining of your lungs, your gut, your liver, the literal borders of your body.
So if I have the flu, which is a respiratory virus attacking my lung lining, IFN-λ can fight it right there.
That's the theory. It ramps up antiviral defenses exactly at the border without sending panic signals to your brain or your muscles.
So you get the protection without the absolute misery.
It defends the castle walls without waking up the king. It's a targeted tactical response rather than carpet bombing the entire city.
This really brings it all back to that group chat idea we started with. It's not just about sending a text message, it's about who receives it.
And exactly how they interpret it.
So stepping back for a second, why does the listener who, might just be driving to work or folding laundry right now, why do they need to know about 4 helix bundles and kinks in a pool noodle? Why does the geometry actually matter to them?
Because we are moving out of the age of just observing biology and into the age of engineering it.
What do you mean by that?
Well, for a long time, we'd say, oh, look, IL-10 stops inflammation. But now that papers like this one by Travella and Polikarpov are mapping the exact atomic structure, the precise angle of the kink in helix F, the V shape of the hug, we can design better keys.
We can 3D print our own text messages.
Exactly. We can design a drug that mimics the IL-10 hug to treat lupus, but maybe it lasts longer in the blood. We can design a molecule that looks like IL-24 to kill a tumor, but is much easier to deliver. We aren't just guessing anymore. We are engineering actual solutions based on the blueprint of the body's own communication network.
It changes how you look at a simple rash or a cold. It's not just sickness. It's a literal breakdown in the group chat.
A very active, very high-stakes group chat where one single typo can cause a disease.
I want to leave everyone with a thought today. We talked about IL-24, the smart signal that magically knows the difference between a cancer cell and a normal cell.
It is an incredible mechanism.
Oh, if our body's already making signals that are that intelligent, what other smart messages are hidden in our DNA that we just haven't decoded yet? What other cures are already floating around inside us just waiting for us to read the text message?
That is exactly the question that keeps us in the lab every single day.
A huge thank you to our expert today for decoding the twisty, helical world of cytokines for us.
My pleasure. It was a lot of fun.
And thank you to you for listening to the Deep Dive Lab. If you want to know what else is happening inside you right now, and trust me, it is a lot, make sure you hit that subscribe button. We will see you next time.